Welcome to the Deep Dive. Today we're diving into, well, a really interesting mix of fields, hydrodynamics, stats, ecology, looking at how we can get smarter about collecting data for conservation. Specifically, we're unpacking a paper called SDM meets eDNA, Optimal Sampling of Environmental DNA to Estimate Species-Environment Relationships in Stream Networks. The lead author is Timothy D. Lambert. Okay, so the problem they're tackling is one you might know if you're in ecology. Species distribution models, SDMs, they're fundamental for mapping where species live. But things get tricky when you want to know how many are their abundance, especially in rivers and streams. The old ways, like capture surveys, they're just too slow, too expensive for the kind of detailed abundance data we really need now.
Exactly, and that's why environmental DNA, or eDNA, has generated so much excitement. You just grab some water, sequence the DNA bits floating in it. It's fast, it's relatively cheap. It really has the potential to change how we gather ecological data on a large scale. But there's a big catch. And the authors call it the inverse problem. How do you take that DNA concentration you measured in your bottle and accurately work backward to figure out the actual number or density of the species upstream that shed it? It's complicated.
Because the water moves, right?
Precisely. As soon as that DNA hits the water, it starts diluting, it gets carried downstream, it breaks down. So the water sample you collect isn't just reflecting what's right there. It's this complex mix from potentially quite a large area upstream.
So the river is basically mixing up the signal, blurring the picture of where the species actually are. Our mission, then, for this deep dive is to figure out how Lambert and Ellner developed a way to, well, unscramble that signal. They wanted to move past just guessing where to sample. Their big idea was using statistical optimization, informed by those very water dynamics, to pinpoint the best sampling locations for maximum precision.
Okay, yeah, let's get into their method because it really is quite clever. They created this linear hierarchical model. Think of it like two connected systems. The first system is basically an SDM. It predicts how much eDNA is likely produced locally based on environmental factors, stream temperature, flow, habitat type, things like that. The second system is a mechanistic tracer model. They use one called the eDNA model, which simulates how that produced eDNA actually moves, how it dilutes, travels downstream, and decays over time.
And the really new part, as I understand it, isn't just accounting for the flow and decay. Others have tried that. It's that they specifically model these spatially correlated concentration errors.
Yeah.
Okay, that sounds important. What exactly does that mean? Spatially correlated errors.
It means that if you take two samples, say downstream from the same big tributary, the errors in those measurements aren't independent because they're drawing on some of the same source water, some of the same mixed DNA. Their mistakes, if you like, are related. They're correlated because of the river network's structure.
Okay, so ignoring that would be like pretending you got two completely separate pieces of information when actually they overlap quite a bit.
Exactly. It's a statistical no-no. You'd overestimate how much you actually learned from that second sample. What Lambert and Ellner did was build the network's hydrology right into the statistical model, so it correctly predicts how correlated those errors should be based on how the sampling points are connected upstream.
And they showed how crucial this is, right? They compared their model to a simpler one.
Yeah, they ran simulations. They compared their dynamic model against what they called a null model, one that basically ignored the water flow and just assumed the DNA came from right where the sample was taken. And the results were stark. The null model, it gave really biased estimates of the species-environment relationships. And maybe worse, it seriously overestimated the natural variation in local density.
Whereas their dynamic model?
Their model produced consistent, reliable estimates. It successfully accounted for the transport and mixing effects.
Okay, so if the model works, if it can give unbiased estimates, the next step is using it to plan better surveys.
Yeah.
That's where the de-optimality comes in.
That's right. They weren't just aiming for a decent sampling design. They were searching for the de-optimal design.
Right, de-optimality. For those of us not deep in statistical design theory every day, what's the practical meaning of that?
Fair enough. It's basically a way to find the sample locations that pack the most punch, information-wise. De-optimality focuses on maximizing the precision of your parameter estimates in this case. The parameters describing the species-environment link and its variability. So the question becomes, given a fixed number of samples, say 50 bottles, where exactly in this river network should we put them to learn the most about that relationship? Got it. Maximum information for your sampling effort. So they tested this on simulated river networks.
Yep, 60 simulated networks, these things called optimal channel networks, or OCNs, and they tried it out for species whose distribution was driven by different things, geographic coordinates, stream size or discharge, and also just random environmental factors.
And how did the de-optimal design stack up against, say, just spreading samples out evenly, which seems like a reasonable approach? That's what they called the benchmark designs. Sensible, even spacing, but ignoring the detailed flow physics and the specific patterns of the environmental factors. And the difference was, well, pretty dramatic. The optimal designs consistently outperformed the benchmarks by a lot.
Okay, give us the headline number. How much better?
This is the key takeaway for anyone planning fieldwork. The optimal designs needed, on average, 41.5% fewer samples to get the same level of estimation precision as the benchmark designs.
Wow. 41.5% fewer. I mean, think about that. That's potentially cutting your field and lab costs almost in half.
Absolutely. It makes bigger, more ambitious monitoring projects suddenly seem much more feasible.
And this wasn't just for one type of species niche.
No, the advantage held up across all the different environmental scenarios they tested: geography, stream size, random factors. It confirmed that being smart about sample placement using the model pays off consistently.
So this leads us to the really practical bit, the rules of thumb they extracted. What did these optimal designs tend to look like? Where did the model tell them to sample?
Okay, yeah, there were some very consistent patterns. Rule number 1, position downstream. Optimal samples were almost always located near the very downstream end of a stream reach. On average, they were placed only about 13% of the way upstream from where that reach ended and flowed into the next section.
Okay, that makes intuitive sense. You're catching the signal produced in that reach just before it gets totally diluted and mixed in the next bigger bit of river. Maximize the local signal.
That seems to be the logic, yeah. Maximize the concentration from that specific reach. Rule number 2, aim for purity, usually. Now, this depended a bit on the environmental factor being studied, but for things like geographic position or random covariates that weren't directly tied to river size, the optimal samples tended to come from smaller streams high up in the watershed. Low Strahler order streams, technically.
Why smaller streams?
Purity of the signal. In those small headwater streams, the water, and therefore the eDNA, is likely coming from a relatively small, environmentally uniform area. The signal isn't as mixed or complicated.
But what if the species you care about, or the environmental factor driving it, is really associated with big rivers? You can't only sample the little streams then.
Exactly. And that brings us to rule number 3. Balance, purity, and coverage. While those pure headwater signals are great statistically, if you're studying something like the effect of stream discharge itself, which obviously varies from tiny trickles to massive rivers, you have to sample across that whole range. That means deliberately placing some samples in the large main stem reaches, even though the eDNA signal there is much more mixed, coming from many diverse upstream sources. You need those points to capture the full environmental gradient.
So it's a trade-off. You need the pure samples for clean signal, but you need the mixed, large river samples to cover the full range of conditions you're studying.
Precisely. And that ties into the final rule. Rule number 4, maximize covariate coverage. Across the board, the optimal designs did a fantastic job of sampling the full spectrum of the environmental variable being studied. They tended to sample the extremes, the smallest and largest streams, the highest and lowest values of whatever factor, more often than just a random spread would. They found the optimal design stretched across almost double the range of environmental conditions compared to random sampling.
Because you need those extreme points to really pin down the slope of the relationship, the regression line.
You got it. You need to see how the species responds at both ends of the environmental spectrum.
Okay, so that tells us where to sample. Did they look at which kinds of river networks make this whole eDNA estimation process easier or harder?
Yes, they did. And interestingly, they found that just 2 key network properties explained over 90% of the variation in how accurate the estimation was.
Only 2. What were they?
First, simply the number of reaches in the network. Basically, how many distinct stream segments there are. Think of it as how many independent experiments or examples of the species-environment relationship the network offers. More reaches, more data points, better estimate.
Okay, number of segments. What's the second factor?
The second one they called the total decay length. This is a bit more complex. It combines the network's physical size, the speed of the water flow, and how quickly the eDNA itself decays. Essentially, it measures how much unique upstream area you can effectively see with your samples before the DNA signal fades away completely.
So, putting those together, where's eDNA likely to work best?
Well, the conclusion is that you'll get the most precise estimates in river networks that have lots of distinct reaches, are physically large, have slow-flowing water, and where eDNA decays or settles out relatively quickly. All those factors tend to limit how far the DNA travels and how much it mixes, keeping the signal more localized and easier to interpret.
That makes sense. Now let's talk about replicates. If you're taking multiple samples, should you cluster them together in one spot or spread them far apart? The paper had something interesting to say about that, tied to error types.
It did. The best strategy, clustering versus spreading, depends critically on where your main source of error is coming from. Scenario 1. If your biggest uncertainty is measurement error, maybe your lab analysis has some variability, or there are inhibitors in the water messing with the PCR, then the model suggests clustering your replicates.
Why clustering in that case?
Because taking multiple samples right next to each other helps you average out that measurement noise and get a more reliable estimate of the true eDNA concentration at that specific location.
Okay, but what if you have a really accurate lab process? You trust your measurement numbers.
Right. Scenario 2, if measurement error is small compared to the natural patchiness or local variation in species density, or what they call sigma sure, then the optimal strategy flips. In that case, you should spread your samples out across many different reaches. You're no longer trying to nail the concentration at one spot. You're trying to capture as many independent examples of that natural environmental and biological variation as possible.
So, understanding your error sources lab versus natural variability is key to deciding how to place replicates, and this model helps predict those spatial correlations that influence this.
Exactly. That ability to account for the spatially correlated errors driven by the river network is what allows for this more nuanced, optimized sampling design. It's a big step beyond older approaches.
This really underscores the power of combining these different fields, ecology, hydrology, statistics. It moves eDNA surveys from being, well, maybe a bit of a guessing game to a much more quantitatively rigorous process. The efficiency gains they showed, that 41.5% reduction, are massive, and the rules of thumb provide really concrete guidance for people planning surveys.
They definitely do. But, we should also touch on the limitations, just for context. The model, to make the de-optimality calculations feasible, had to make some simplifying assumptions.
Such as?
Well, for instance, they assumed the measurement errors were constant and followed a nice, simple Gaussian distribution. In reality, eDNA measurement error can be more complicated, often depending on the concentration itself. And crucially, they had to assume that the parameters within the tracer model, things like the exact rate of DNA decay or the flow speeds, were known perfectly.
Which in the real world they usually aren't, right? There's uncertainty in those physical parameters too.
Exactly. You usually only have estimates of decay rates or flow dynamics. So future work probably needs to figure out how to incorporate uncertainty about the physical processes themselves into the optimization.
That makes sense. Handling multiple layers of uncertainty.
It's the next challenge.
So thinking about the future for you, the listener, perhaps planning eDNA work. The paper ends on a really intriguing note about adaptive sampling. What's that about?
Yeah, this is a cool idea. They suggest that maybe the absolute best approach isn't to create one fixed sampling plan up front. Instead, you could use an adaptive sampling scheme. You'd go out, collect an initial batch of samples, run the analysis.
And use those first results to refine things.
Precisely. You'd use that initial data to get better estimates of your error variances, maybe figure out which environmental factors seem most important for your specific species and system, and then you feed that updated information back into the optimization model to intelligently choose where to put your next round of samples.
So sampling becomes a dynamic process, learning and refining as you go, not just a one-shot plan.
Exactly. It emphasizes that optimal sampling might be less about a static map and more about an ongoing cycle of measurement, modeling, and refinement. That's a pretty exciting direction for ecological monitoring, integrating the fieldwork and the analysis much more tightly in real time.